Lapel.
From lab to life, medicinal chemistry behind PARP-1 inhibitors. Hey everyone! Welcome back to our channel. Today, we're diving into the fascinating world of medicinal chemistry, focusing on the structure-activity relationship, SAR, of PARP-1 inhibitors. If you've ever wondered how scientists design drugs to target cancer, you're in the right place. We're excited to explore the intriguing realm of cancer therapy, specifically how PARP-1 inhibitors are shaping the future of treatment. Recent advancements in the structural design of these inhibitors highlight their potential as powerful anti-cancer agents. Poly-A-D-pyribose, polymerase 1, PARP-1, plays a crucial role in repairing DNA damage, and inhibiting this enzyme can lead to synthetic lethality in cancer cells, particularly those with DNA repair deficiencies, like BRCA1 and BRCA2 mutations. Let's break down how this works and what it means for cancer therapy. First off, PARP-1 is a protein that plays a crucial role in repairing DNA damage in our cells. When DNA gets damaged, PARP-1 jumps into action, helping to fix those breaks. However, in cancer cells, this repair mechanism can actually help the cancer survive. That's where PARP-1 inhibitors come into play. Now, let's talk about the structural types of these inhibitors. Most PARP-1 inhibitors share a common feature: they contain an amide group. This group is essential because it allows these inhibitors to compete with NAD+, a molecule that PARP-1 uses to perform its repair functions. By blocking this interaction, the inhibitors prevent PARP-1 from doing its job, leading to the death of cancer cells. But that's not all. Recent research has also led to the development of non-amide PARP-1 inhibitors. These compounds don't contain the traditional amide group but still effectively inhibit PARP-1. This opens up exciting possibilities for creating new drugs with potentially fewer side effects. Now, let's discuss some key characteristics of PARP-1 inhibitors. One important aspect is their potency. For instance, some of the latest inhibitors are effective at low nanomolar concentrations, which means they can work effectively even in very small amounts. This is a huge advantage in minimizing side effects for patients. Another characteristic is selectivity. The best PARP-1 inhibitors are designed to specifically target PARP-1 without affecting other members of the PARP family. This selectivity is crucial because it reduces the risk of damaging normal cells, which can lead to unwanted side effects. Speaking of effectiveness, let's take a look at some PARP-1 inhibitors that are currently in clinical studies. The FDA has approved several, including olaparib, rucaparib, niraparib, and talazoparib. These drugs have shown promising results in treating cancers like ovarian and breast cancer by significantly prolonging progression-free survival for patients. And there's more. Veliparib is also in late-stage clinical trials, adding to the growing list of PARP-1 inhibitors that could soon be available to patients. As we look to the future, researchers are exploring new structural designs and combinations of PARP-1 inhibitors with other therapies. This could enhance their effectiveness and broaden their applications in treating various cancers. Now, what do we mean by structure-activity relationship? In simple terms, SAR is the relationship between the chemical structure of a compound and its biological activity. Understanding this relationship helps researchers modify drug structures to improve their effectiveness and reduce side effects. Here's where it gets interesting. Recent studies have shown that many PARP-1 inhibitors share common structural features. For instance, most of them contain an amide group. This group is crucial because it mimics the natural substrate of PARP-1, allowing the inhibitor to effectively bind to the enzyme and block its activity. But it doesn't stop there. Researchers have discovered that the presence of certain functional groups, like halogens, can enhance the potency of these inhibitors. For example, adding a fluorine atom can increase the lipophilicity of the molecule, which helps it penetrate cell membranes more easily. Now, let's talk about ring structures. Studies suggest that 6-membered ring systems tend to exhibit better activity than 7-membered rings. Why is that? Well, it's all about how these rings interact with the target enzyme. The right shape and size can make a huge difference in how well the drug binds to PARP-1. And here's a fun fact: not all PARP-1 inhibitors need to contain the traditional amide group. Some novel compounds, known as non-amide inhibitors, have been developed that still effectively target PARP-1. This opens up new avenues for drug design, allowing scientists to explore a wider range of structures and mechanisms. So, what does this mean for cancer treatment? By understanding the SAR of PARP-1 inhibitors, researchers can design drugs that are not only more effective but also have fewer side effects. This is crucial in the fight against cancer, where patient quality of life is just as important as treatment efficacy. In conclusion, the structure-activity relationship of PARP-1 inhibitors is a dynamic and evolving field. As scientists continue to uncover the intricate details of how these drugs work, we can expect to see more targeted and effective therapies in the future. Let's move on to cancer therapy and PARP-1 inhibitors. So, first things first, what exactly are PARP-1 inhibitors? PARP stands for poly-A-D-pyribose polymerase, which is an enzyme that plays a crucial role in repairing DNA damage in our cells. When DNA gets damaged, PARP-1 jumps into action to help fix it. However, cancer cells often rely on this repair mechanism to survive and grow. That's where PARP-1 inhibitors come in. These drugs block the activity of PARP-1, preventing cancer cells from repairing their DNA. As a result, the cancer cells accumulate damage and eventually die off. This process is known as synthetic lethality, where the combination of a PARP inhibitor and a defect in DNA repair pathways, like those seen in BRCA mutant cancers, leads to cell death. Now, let's talk about some of the PARP-1 inhibitors that have made their way into clinical practice. The FDA has approved 4 main ones: olaparib, rucaparib, niraparib, and talazoparib. Each of these drugs has shown significant effectiveness, particularly in treating cancers like ovarian and breast cancer that are associated with BRCA mutations. For example, olaparib was the first PARP inhibitor approved and is used for patients with ovarian cancer. It's an oral medication, making it convenient for patients. Niraparib, on the other hand, is often used as a maintenance therapy after initial treatment, helping to keep cancer at bay. So, how do these inhibitors work at a molecular level? Well, PARP-1 inhibitors bind to the enzyme's active site, which prevents it from attaching to DNA and doing its job. This trapping of PARP-1 on DNA creates what we call toxic lesions. These lesions are so severe that they hinder the cancer cell's ability to divide and grow, leading to their eventual death. It's important to note that while PARP-1 inhibitors are effective, they can also affect normal cells. This is because normal cells also rely on DNA repair mechanisms, albeit to a lesser extent. Therefore, researchers are constantly working on improving the selectivity and reducing the side effects of these inhibitors. Now, let's explore the therapeutic implications of PARP-1 inhibitors. Beyond just treating BRCA mutant cancers, there's growing evidence that these inhibitors could be effective in other types of cancers as well. For instance, studies are looking into their use in triple-negative breast cancer and pancreatic cancer, which are notoriously difficult to treat. Moreover, researchers are investigating combination therapies, using PARP-1 inhibitors alongside other treatments like chemotherapy or immunotherapy. This approach could enhance the overall effectiveness and help overcome resistance that some tumors develop. Looking to the future, there's a strong push for developing new, more potent PARP-1 inhibitors that are less toxic and more effective. Scientists are exploring non-amide compounds, which could provide a broader range of therapeutic options. PARP-1 inhibitors represent a promising frontier in cancer treatment, leveraging the unique vulnerabilities of cancer cells. Their ability to induce cell death in tumors with DNA repair deficiencies opens up new avenues for therapy. Now let's talk about the future of PARP inhibitors. First, let's recap what PARP inhibitors are. PARP, or poly-A-D-pyribose polymerase, is a protein that plays a crucial role in repairing DNA damage in our cells. When cancer cells have damaged DNA, they often rely on PARP to fix it. By inhibiting this protein, we can effectively starve cancer cells of their repair mechanism, leading to their death. Pretty cool, right? Now, as we look to the future, there are a few key points to consider. researchers are really focused on improving the effectiveness and safety of these drugs. While we currently have 4 FDA-approved PARP inhibitors, olaparib, rucaparib, niraparib, and talazoparib, there's still a lot of room for improvement. One of the major challenges we face is the side effects. Many PARP inhibitors can cause cumulative damage to normal tissues, which isn't ideal. So, enhancing the selectivity of these drugs, meaning they target cancer cells more effectively while sparing healthy cells, is a top priority. Researchers are also exploring new structural types of PARP inhibitors. By modifying the chemical structures, they hope to discover new compounds that are not only more effective but also have better solubility and longer half-lives. This means patients could take them less frequently, improving compliance and overall treatment experiences. Another exciting avenue is the use of computer-aided drug design, or CADD. This innovative approach allows scientists to simulate and predict how new drug compounds will interact with PARP proteins before they even enter the lab. It's like having a sneak peek at which compounds might work best. Moreover, as we gather more data from clinical trials, we'll be able to tailor treatments to individual patients better. This personalized medicine approach is the future of cancer therapy, allowing us to match the right drug to the right patient based on their unique genetic makeup. In conclusion, while we've made significant strides with PARP inhibitors, the journey doesn't stop here. The ongoing research and development in this field hold great promise for improving cancer treatment outcomes. With continued innovation, we could see even more effective therapies that not only fight cancer but also enhance the quality of life for patients. Thanks for tuning in today. If you found this information helpful, don't forget to like and subscribe for more insights into the world of medicine and cancer research. Until next time, stay curious.
PharmaPal.